Hello, good morning again. Today we are going to discuss Unit 5 of your course, that is MFN 003 Microbiology, and the topic today of discussion is food hazard and your microbial origin. We will start with the meaning of a foodborne disease. What do we understand by the foodborne disease? Foodborne disease is the outbreak is defined as an incident in which two or more persons experience a similar illness, usually gastrointestinal, after the ingestion of a common food which is identified as the source of foodborne illness. Okay, so after that we have types of your foodborne diseases. There are various types of foodborne diseases. We have food intoxication, food infection, and foodborne toxic infection. Okay, food intoxication. It involves the toxic substances produced in the food before it is consumed. Produced in the food by microorganism before it is consumed. The toxins in the food makes the person sick. For example, you have Staphylococcus aureus food poisoning. Next, we have your food infections. It is ingestion of food containing bacteria and then growth of bacteria in the body. Example, you have salmonellosis caused by the salmonella bacteria. It generally occurs when you are having or when you are consuming chicken and egg or any sort of poultry. Okay. Foodborne toxic infection, they are caused by the ingestion of a large number of enterotoxigenic strains of bacteria which, while multiplying in the intestine, produce and release enterotoxin in the intestine. So these are your differences between your food intoxication, food infection and foodborne toxic infection. Okay, you can give the definition and then you can write down the what kind of example of a microorganism that is causing each kind of disease in the body. So that will do it. Next, elaborating on your food intoxication. It occurs in protein rich food. Okay, bacteria which has a good growth temperature of 35 to 37 degrees Celsius, which is basically your normal temperature or your room temperature. Okay, such bacteria, they have a growth range from 7 degrees Celsius to 48 degrees Celsius. So it is very difficult to contain or to stop the bacterial growth for this sort of bacteria, because at normal temperature anything can grow. Okay, so you have we will discuss two main type of bacteria, that is Staphylococcus aureus. These are poisoning due to enterotoxin which are heat stable. Preventive measures, you can go through the preventive measure in page number 93. It causes the inflammation of your intestine lining. Okay, next we have your Bacillus aureus poisoning. Growth temperature is from 20 to 30 degrees Celsius. Again, it is within your normal room temperature. And 10 to 45 degrees, it is spore forming, can survive severe heat processes, normal cooking temperature, and does not kill the spore. That means normal heating temperature or your normal cooking temperature is not going to kill the spore, because Bacillus aureus is a spore forming bacteria. However you cook it, the spore that is there, it is going to be resistant to the heat. It will be staying in the food, and after the ingestion of your food, the spore in your body will then starts to incubate, and then it will lead to the further growth of your Bacillus aureus in the body. Okay, it causes diarrhea. Other strain causes nausea and vomiting immediately after 2 to 5 hours of food ingestion. They are generally found in soil, vegetables, grains, cereals, dairy products. Preventive measures for this can be discussed in page number 95. Okay. Next, we are going to discuss one of the most severe, but it is rare condition, that is your botulism. Okay, botulism means a serious neurological illness. Causes are the improperly preserved canned foods containing neurotoxin by Clostridium botulinum. So this is a microorganism that is causing your botulism. Okay, C. botulism are capable of slow growth and produce toxin at low temperatures, that is 33 degrees Celsius. 8 types of toxins are known right now. It is A, B, C1, C2, D, E, F and G. Some strain produced mixed toxins. Okay, so it is very difficult to treat it. Okay, so as early as diagnosed, it has to be treated. Found, it is distributed in soil and marine sediments throughout the world. 2. Intestinal tract of animals including fish. Okay, outbreak, product of fish, marine animal, meat, poultry, fruits and vegetables including mushrooms. Okay, so whenever you are eating your canned food, or if it is properly not, certain measures has to be taken for the canned food to be processed. Okay, so if it is not, food safety standards is not followed, then that can lead to your botulism. Treatment, as it is page number 96. You can go through the treatment in page number 96 of your unit of your book or your handout. So moving on to your foodborne infections, you have salmonellosis and shigellosis. Okay, salmonellosis is a very common. So talking about salmonellosis, it is a very common, disease or that occurs in the human who generally consume poultry and egg most of the time. So it because the growth temperature of the salmonella is around 38 degrees Celsius. Okay, precautionary step that is to be maintained, page number 99. I'll just read it out. You have to check the checking of the animal to be free from contamination, avoiding cross-contamination from raw to cooked foods, destruction of salmonella by proper cooking. How do you destroy salmonella? It's a pasteurization and heat treatment. That's 60 degrees Celsius in 15 to 20 minutes. That is the heat treatment that is done to the properly removing of salmonella. Cooking the prepared food rapidly to below 70 degrees Celsius and storing within the refrigerators or freezers or held at a temperature above 60 degrees Celsius. Removing food handlers who are carriers from the food preparation area and improving general sanitary conditions in the premises and ensuring better pest control. So this is the salmonellosis. Next we move on to shigellosis. Shigellosis is causes bacillary dysentery. Okay, growth at 37 degrees Celsius with a growth range starting from 10 degrees Celsius to 40 degrees Celsius. Okay, they are heat sensitive. They cause nausea, diarrhea. Page number 99. You can go through the steps that can be help to minimize the problem of your shigellosis. Okay, next we have your Escherichia coli diarrhea, hepatitis A, shellfish poisoning and Vibrio myelitis gastroenteritis. So you can just read through it. So basically it is the same. When the question is you will be questioned to describe any 2 or 3, okay, or just write down the name or the type of foodborne infection. So you can just read through it. Next we go to your foodborne toxic infection. Under foodborne toxic infections, we have already discussed the definition. What is the definition of foodborne? We have Clostridium perfringens gastroenteritis. Okay, it it is caused by the organism Clostridium perfringens. Okay, it can withstand boiling temperature for 6 hours. 43 degrees Celsius to 45 degrees Celsius is the growth temperature. Vegetative spore destroyed at temperature 60 degree and above. Okay, the vegetative spore of the Clostridium perfringens is destroyed at a temperature of 60 degree and above. Strain of C. perfringens, that is A, spores survive heat resistant. They survive till at 95 to 100 degrees Celsius for 1 hour. But if you heat it beyond that temperature, then the spore is generally going to deteriorate, or it is not going to survive. Okay, they produce toxins on food after ingestion and they starts to sporulate. Mild food poisoning is caused by the Clostridium perfringens organism. Okay, and next, like this, we have enterotoxigenic Escherichia coli, which causes a gastroenteritis. We have cholera, which is caused by Vibrio cholera, and listeriosis. Okay, now you can read it in your copy. Make short notes like this, what like Clostridium perfringens, not to explain in details. You have to name the organism that is causing it, the growth, how to which growth range it can be grown. Okay, what are the preventive measures that can be applied to prevent the infection, the toxic infection in the body. So very easy. Now next we move on to your mycotoxins. So what are mycotoxins? Mycotoxins are substances produced by moles like Aspergillus flavors, etc., produced in agricultural commodities either during pre or post harvest stages, which may be toxic or produce adverse effects in the living organism. They are found in agricultural commodities like cereals, example maize, wheat, oilseeds, example groundnuts, and spices in chilies, etc. Okay, poisoning caused by mycotoxins is called your mycotoxicosis. Okay, so these are this is your mycotoxins. Okay, so how will you be able to prevent or remove the mycotoxins? So you can read it from your book, or I can just read it out. Just a second. So we have types of mycotoxins. We have aflatoxicosis. Next we have your deoxynivalenol mycotoxicosis, and then you have ergotism. Ergotism, I think it's generally very common, and I have seen ergotism. It is a disease of cereal crops like rye, wheat, millet, sorghum, and grasses. The crops, they become black in color. Okay, so that is your ergotism. Okay, poisoning attributed to ergot of rye and therefore referred to as ergotism. Okay, next we have your foodborne diseases due to naturally occurring toxicants in the food. A variety of naturally occurring food toxins have been linked to human ill health and death. Among them, the principal toxicants that have been responsible to cause disease outbreaks in humans in India include the following: laterism, veno-occlusive disease, and epidemic dropsy. So these are these three types of your outbreaks. Okay, so we'll go through laterism in short. Laterism is caused by the overconsumption of keshari dal. Okay, generally happens in Karnataka, Maharashtra, and Andhra Pradesh, because keshari dal is a very drought resistant legume, and when there is matters of drought, people tend to consume more of the keshari dal. Okay, so what causes laterism? Keshari dal contains neurotoxin amino acid beta oxalyl amino alanine, BOAA. Okay, this results in the crippling of paralysis of lower limbs, because the body is not able to break down this particular amino acid. It gets accumulated and further leads to your paralysis. Next we have your veno-occlusive disease. It is or VOD. VOD of liver. It is a toxic liver injury caused by the pyrrolexidine group of alkaloids. Okay, then we have a epidemic dropsy. It is a toxic. It contains toxic alkaloid sanguinarine, which interferes with the oxidation of pyruvic acid, which accumulates in your blood. So these are all either they are alkaline or either they are alkaloids. They are hampering in the metabolism process of the body, and hence it is it is getting accumulated in the body, and then later it cannot be removed. So it causes these certain type of disorders in the body. Okay. Epidemic dropsy, which food? If you talk about food in dropsy, basically arginine oil. Okay, arginine oil. When a mustard oil is contaminated with arginine oil, so arginine oil is the culprit, because arginine oil consists of this toxic alkaloid sanguinarine. Okay, so consumption of mustard oil or consumption of mustard oil adulterated with arginine oil, or consumption of arginine oil alone leads to your epidemic dropsy. Okay, so vegetable oils, when it is being bought or when it is being processed, it should be very much taken into consideration that what has been put into the vegetable oil. Okay, so as to check whether there is no adulterant present. Reporting and investigation of foodborne diseases. So reporting, and it is, if you see and investigate an outbreak, then you receipt of an initial data, you determine of whether an outbreak has occurred, description of cases in terms of time, place and person, epidemiological investigation, formulation of hypothesis and laboratory studies, and synthesis of findings with conclusions and recommendations. Okay, so this is what it is done. Okay. Certain additional factors which needs to be considered include the number of people possibly affected, the severity of the suspected disease, time for which illness occurred, and the potential for continued disease transmission. Okay, after that you need to plan an investigation. Then you have a demographic information, food history, data screening. Okay, so this all comes under your investigation of an epidemic outbreak. Then you have epidemiological analysis, which consists of your time, place, person, other important information. Then we have D, we have analytical epidemiological analysis. Okay, so table 5.1. This is the table which is showing you the food specific attack risk at a marriage party. It is an example is taken, like for example, a epidemic occurs in a marriage party, or there's an outbreak in a marriage party. So how will you handle it? Okay, what was the food served? How many people are affected? How many people are not affected? Okay. Then obviously there is a collection of sample. Then you have laboratory analysis. Okay, so in your practicals, in your food science practical, you will be doing this adulteration practicals. So that will help you a lot in order to identify whether there are certain adulterants present in the food or not. So more will be clear to you when you actually do the practical. Okay, and here you have something known as glossary in page number 119.5.10. So you can go through the glossary. You can you can check your progress exercises in your book. Answers are already given, but I rather suggest you to do the progress check before and then tally with the answers. So for that, today we come to the end of unit 5. In short, I hope you understand whatever has been taught to you. If you do not understand, you can always go through your book. You can go through net. Okay, so thank you. Next we will discuss unit 8 in our next class.